Okay, to understand biochemistry, we need to first know some basic terms. So these basic terms are going to be functional groups, sorry, functional groups, monomers versus polymers, isomerism, yeah, it's the three terms. So what functional groups are, you can think of them as polyatomic ions, except not necessarily with a charge. So they're groups of atoms, so many atoms, that act as one. So the most prominent functional groups in biochemistry are going to be a hydroxyl or alcohol group, which is an OH connected to a larger chemical. We also have the carboxyl or carboxylic acid group, which is this, and it can sometimes act as an acid because that H can get loose at times. You also have an amine group, which is a nitrogen connected to two hydrogens connected to something else. You have a methyl group, which is a C connected to three H's. A phosphate group, which is an O, P, O with a negative charge. And then lastly, you have a sulfhydryl group. Now, the sulfhydryl group you're not really going to see as much, but where you're going to find it is on large carbon chains. So let's say we have a carbon chain, and you'd find sulfhydryl groups connected to these. And typically they're more carbon chains. So this results in the sulfhydros sort of locking onto each other, and this produces very durable materials such as rubber. Now let's move on. So monomers versus polymers. So, sorry for the misspell. So monomers and polymers, you can think of monomers as the most basic building blocks. So polymers are just many monomers that are linked together to form a greater chemical. And then isomerism is when you have the same chemical formula. So same formula, different structure. So I'll show you a very prominent example of isomerism. It's the most basic type. So it's called structural isomerism, which all that means is you have the same formula, just slightly different structure. So these are all carbohydrates. We're going to talk about that later in the video. But if you count up all the atoms, you'll notice that the formula for all of them are C6H12O6. And, you can see that some of them are just subtly different from each other. So like this, look at here, galactose and glucose. The only difference really is the position of the H and OH. On galactose, it's this way, while in glucose, it's flipped. But that's enough of a change to make it its own chemical. Next, you have geometric isomers, which are these. So here you see you have this, both of these are on this side, while here they're on opposite sides. And lastly, we have enantiomers or optical isomers. And you can think of these as mirror images of each other. So it's like you have a mirror here, and these are mirror images. So since they're mirror images, you can never quite rotate them or shape them in any way to make them quite the same. So you can't like fit one on top of the other to make it identical. And an interesting property, which is why they're called optical isomers, is they rotate polarized light in opposite directions, which means that they rotate light that only comes in a specific direction opposite ways. So you have L and D isomers. So we have L and D isomers, and L isomers rotate light left, and D isomers rotate light right. Now, an interesting fact is that L isomers only in amino acids, which are found in biological systems. So amino acids only have L isomers and never have D isomers, although that's not true for all biological beings. So like, sure, only amino acids might only have L isomers, but that doesn't mean that D isomers can't be found in any living system. So now that we're done with these basic terms, we can move on to talk about carbohydrates. Now, carbohydrates are also known by another name. We call them simple sugars. And you can see these are some examples of carbohydrates. So for all intents and purposes here, we're just going to say that the most simple a carbohydrate can get is C6H12O6, which means that you either have glucose, galactose, or fructose. Now, I'll pull up the chain image. This is a chain form of a carbohydrate, but it's not always like this in nature. You can also have it in a ring form, which looks like this. And all this is you have a slight change. You basically fold the molecule in so it forms the structure, and you just assume that at these intersections there's carbons. Now, for our case, this is simple sugar, and this is a monosaccharide, which means that this is a monomer in terms of carbohydrates. And you might be asking, how do we form polymers? So what happens is we have, let's see if my computer would cooperate. Let's say we have two of these. So we have two monosaccharides. So what's going to happen is an oxygen, so let's just say this oxygen, and an alcohol group are going to combine. So they're going to combine. So what happens is that you take the oxygen and a hydroxyl group, and it forms an H2O. Now, once you have the H2O removed, what happens next is that the oxygen over here, it moves and comes to connect the two molecules. And this is an oxygen bridge. More properly, it's referred to as a glycosidic bond. And the process where you're losing a water molecule between the molecules, to form a bond is called dehydration synthesis. It's also known as condensation reaction. Now, what we see here is a disaccharide, so just two sugars. Now, the process doesn't stop here. You can, have the disaccharide bond with, even more monomers or disaccharides and whatnot, and you can have a polysaccharide. So you can end up with many sugars, all linked together in one atom. Now, while you're having molecules undergo dehydration synthesis, it's important to keep in mind that you're not doubling the formula. So you can't just do this because you would end up with this. So if you double C6H12O6, you would end up with C12H24O12. And that's not it, because we lost a water molecule. So what you're going to do is subtract the water molecule, and you're going to end up with C12H22O11. Now note that the hydrogen will always be in a 2-to-1 ratio with the oxygen. So no matter what, it's going to be in 2-to-1 ratio, hydrogen to oxygen. And that's actually a good way to identify simple sugar as carbohydrates, because you see these carbons and you see them surrounded with an H and an OH. And that's really useful for knowing the name, because you can think of carbohydrates as just a chain of carbons, and they've been hydrated. You've added waters to them, so you put an H on one side and put the OH on the other side. Now later on, as we go over lipids, keep in mind that not all substances with the HCOH are carbohydrates, but it's just useful when you have to identify things. Now, carbohydrates have a variety of uses. The most notable one is just immediate energy. So carbohydrates can be converted into energy. So for us humans, we just convert glucose. So this can just undergo cellular respiration and we convert it into energy. So, but that's not the only thing we can do with these. So if we bond a lot of these together, we can turn it into a sort of intermediate energy storage. And now the reason why I'm calling it intermediate is because there's longer term energy storage methods. So the shortest term, well, aside from ATP, which we're not going to touch on right now, is glucose. So just a monosaccharide. So glucose is the lowest, so it's just immediate energy. And lipids, so fats, are used for very long-term storage. So things like polysaccharides, so stuff like starch, cellulose, they would fall somewhere in the middle. And lastly, you also can use carbohydrates as structural support. So certain living things, such as crustaceans and some insects, they use a carbohydrate called chitin, and they just use it as for an exoskeleton, so to provide structure.